Hello, we are Group 3 and our proposed topic is entitled Serine Protease Inhibitory and Antioxidant Activity Between Different Preparations of Coffea canephora and Coffea arabica beans. In the Philippines, there is an increasing demand for coffee, with Coffea arabica and Coffea canephora being two of the most popular beans being brewed locally. Bioactive compounds present in coffee beans have garnered considerable attention for their proposed health benefits, especially their antioxidant activity. So brewing methods and or preparations can actually affect their free radical scavenging activity or their antioxidant activity. And on a similar note, serine protease inhibition is associated with these antioxidant activity as it tests the inflammatory response that often generates free radicals. Studies delving on both the serine protease inhibition and antioxidant activity of these locally produced coffee beans are actually limited. Hence, it remains a compelling idea for research. So, the main objective of the study is to determine and compare the serine protease and the free radical scavenging activity between the two different preparations, brewed and espresso, of Coffea canephora and Coffea arabica. Specifically, this study aims to determine and compare the free radical scavenging activity of brewed and espresso preparations of Coffea canephora and Coffea arabica using DPPH. And next is to determine and compare the serine protease inhibitory activity of Coffea canephora and Coffea arabica in brewed and espresso preparations.
The theoretical framework of the study is divided by its independent and dependent variables and their methods of testing. So the independent variables are your two coffee preparations, brewed and espresso, Coffea canephora and Coffea arabica. They are subject to DPPH assay to find the free radical scavenging activity or their antioxidant activity, and serine protease inhibitor assay to find the serine protease activity. The study presents with three null hypotheses. The first is that there is no significant difference in the serine protease inhibitory activity of both coffee preparations, brewed and espresso. The second is that there is no significant difference in the free radical scavenging levels or the antioxidant levels of both preparations. And the third null hypothesis compares in general the serine protease and free radical scavenging activity between the coffee species, Coffea canephora and Coffea arabica.
For our methodology, first, our research design will test the cause and effect relationship between the dependent and independent variables. Second, it will aim to test the free radical scavenging and serine protease activity through DPPH assay and serine protease inhibitor assay respectively. Third, both positive and negative controls will be used to ensure the validity of the experiment. And lastly, results will be measured quantitatively and their statistical significance will be calculated for comparative analysis.
The procedure will begin with a coffee extraction preparation. The two coffee beans will be finely ground and divided into two groups for distinct preparations. The first involves brewing, while the second employs an espresso machine. Then it will be cooled down and filtered. The resulting brews will be utilized for the DPPH assay and protease inhibition test. In a testing well, DPPH solution is combined with GIST HCl buffer. The sample solution, the coffee, is swiftly added to the well and left at room temperature for 30 minutes. The absorbance of the sample is then recorded at 517 nanometer using a UV visible spectrophotometer. For comparison, a blank solution is prepared with ethanol and tris HCl buffer. The inhibition ratio expressed as percentage calculated using the formula inhibition ratio is equal to A1 minus A2 over A1 times 100, where A1 is the absorbance of the blank solution and A2 is the absorbance of the testing sample. The inhibition assay measures how well a sample can inhibit or reduce the activity of free radicals.
For the serine protease inhibitor assay, first, a 1 ml volume of the sample will be mixed with an equal volume of trypsin, and then it will be pre-incubated at 37 degrees Celsius for 15 minutes. And then, another 2 ml of 2% casein solution will be added, and then it will be allowed to stand for 30 minutes at 37 degrees Celsius. And then trichloroacetic acid of 5% solution will be added to stop the reaction, and then it will undergo centrifugation for 15 minutes. After this, the absorbance will be measured at 280 nm. Then this protease inhibitor activity will be expressed as an inhibition percentage, and it will be solved using the previous formula. And with these steps, we will know the protease or serine protease inhibitor activity of the tested samples.
First, in our statistical analysis, results will be presented as the means standard deviation. Second, one-way analysis of variance or ANOVA will be conducted for data analysis. And third, significant difference will be considered at p-value of less than 2 or equal to 0.05.
To present our scope and limitations of the study, our research aims to examine and compare the serine protease and free radical scavenging activity of Coffea canephora and Coffea arabica coffee varieties. We will be focusing on utilizing specific tools, instruments, and equipment necessary for the analysis. The scope of our study will involve comparing the serine protease and free radical scavenging activity of the two coffee varieties, utilizing the DPPH assay to determine the serine protease and free radical scavenging activity, examining two distinct forms of each coffee variety, namely brewed and espresso preparation, to comprehensively analyze the difference in their properties, and lastly, investigating the serine protease activity of both coffee varieties at two preparations, allowing for a comparison. While our study has a defined scope, there are certain limitations to consider, and these limitations include: firstly, the assessment of free radical scavenging activity; secondly, the influence of other factors; and lastly, the availability of samples.
Next, for our significance of the study, the results of this study could be highly beneficial to the following: coffee consumers, to the coffee farms or producers, to the medical industry, to the food industry, to our local community, to our future researchers. Next, for our operational definition of terms, the following terms will be described in the study: antioxidant, the Coffea arabica and Coffea canephora, the two coffee plants that will be utilized in the experiment; the brew and the espresso preparation, the two methods that will be utilized in the experiment; and the other terms are free radical, the serine protease inhibitor assay, and lastly, the 2,2-diphenyl 1-picril hydrazine or DPPH assay.